Today, our topic is regarding the acid base physiology, or we can say that acid base balance. And this lecture is like surface level concepts, it's not like indulging into a deep depth, right? And it will help you to answer the questions regarding the pH and the buffer. Okay, so let's get started. First of all, we are going to discuss about the concentration of hydrogen ions in our body or in our serum, in our blood. Okay, as we know that the sodium concentration ranges from 135 to 145, please concentrate on the units. It's milli equivalent per liters. Okay, and the potassium concentration is 3.5 to 5.5 milli equivalent per liter. But when we talk about the concentration of hydrogen ions, we are dealing with very small number, right? We are dealing like nano equivalents or nano equivalents per liter, because the concentration of protons in normal healthy body is approximately this amount, and it's a very tiny amount. You can see that after the point there are seven zeros. Okay, when we write the number as 0.000000004, these are seven zeros. You need to remember that it's milli equivalent per liter, or we can just write 40 nano equivalent per liter. It's not milli, it's nano, because the hydrogen ion concentration is kept very low, and it is due to the fact that these hydrogen ions or the protons, they maintain the pH of our blood, right? Basically, the concentration of hydrogen ion is equal to the pH of the blood, and now we are exactly deriving that equation. Okay, there was a scientist named as Svensson. He introduced the negative log of this value. It means that he took the negative log of this value. He said that these zeros are very difficult to understand and to deal with the zeros, so he just introduced the negative log of this value, and from here the concept of the pH came, right? So let's talk about how the Svensson introduced the negative log of this value. First of all, these brackets. You need to remember that these brackets show the concentration of any ion inside them, right? For example, if I put H, so it means that hydrogen ion concentration is being represented. So we can say that hydrogen ion concentration is equal to this value. Okay, we know that this value has seven zeros after point. So he introduced logarithm, right? It's very important that we took log on both sides. Now we get the log of hydrogen ion concentration, and when we use the calculator and we put this value and take a log of it, it becomes minus 7.4. Okay, it's minus 7.4. But is it okay to deal with the minus when talking in medical? No, we need to get rid of this minus thing, right? So to get rid of this minus thing, we need to multiply on both sides by minus, because as multiplication, minus multiplied by minus is going to be plus. When we take negative on this side, it's become negative log of hydrogen ion concentration is equal to minus into minus 7.4. So this is our main like you can say equation, in which minus log of hydrogen ion concentration is equal to positive 7.4. But we do not write positive sign in general. Normally we do not write positive sign, right? So just for the sake of convenience or sake of understanding, we write minus log of hydrogen ion concentration is equal to 7.4, and that's exactly what we need. Svensson scientists introduced the negative logarithm of hydrogen ion concentration, which became equal to pH. As you know that our blood basically, pH is approximately 7.35 to 7.45, but now in this lecture we are going to just take the number of 7.4. Okay, now let's see how the word pH is derived. As we know that minus log of hydrogen ion concentration, again we are going to say that minus log of hydrogen ion concentration. No, we need to get rid of this minus log as well, right? So in place of minus log, we can just write power of hydrogen ion concentration is equal to 7.4. But later on, this power was converted to P. Okay, this power was converted to P and H as it is, and 7.4. So our body's pH, our blood pH, is 7.35 to 7.45 in general. If it's high, it's alkalosis, and if it's low, it's acidosis. And that's the condition when the hydrogen ions are fluctuated in our blood, right? So we need to maintain this pH within a normal range. Okay. So normal pH is negative log of hydrogen ion concentration. Okay, this is the negative log of hydrogen ion concentration. But when we say that when the hydrogen ions in the blood increases, the pH will goes down, and when hydrogen ion goes down, it means that pH is going to increase. But how this all calculations are going to be occur? We are discussing about that. Okay, we know that hydrogen ion concentration is normally it's this value, right? But when we increase the hydrogen ions by 10, we are making the blood more acidic. It means that on the pH scale, what number is going to be obtained? It's less than 6, is less than 7, or more than 7? It's less than 7, because anything less than 7 on the pH scale is going to be acidic. When we multiply this number by 10, it means that we are increasing a lot of hydrogen ions in that particular solution or the blood in our case. It means that when we take log on both sides, okay, we are taking log on both sides, that's repeating the all calculations. But when we take the log of this value, it becomes 6.4. Okay. But again, we are going to get rid of this minus as well, right? Like that's we have done, right? So when we take the log of this value, it's going to be less. It's going to be acidic. That's what we need to calculate, that increasing the hydrogen ions drops the pH. And when we decreases the hydrogen ion, it means that when we divide this number by 10, we are decreasing hydrogen ions by 10. So the pH will going to increase. It become 8.4. You can calculate it by your own calculators, right? So when we decrease the hydrogen ion concentration, that's the pH. When it's going to increase, it going to become alkaline, it going to become basic. When we increase the hydrogen ion concentration, the pH going to decrease, it going to be acidic, it's going to be less than 7.4. Okay, driving this equation, we need to understand that in our body a lot of acids are being produced. But how? Because a lot of metabolism is going on there, right? In our all cells that are metabolically active, they are constantly producing carbon dioxide as a byproduct of cellular respiration, right? We study in biochemistry that glucose plus oxygen combines and they form carbon dioxide and water, right? And a lot of oxidative phosphorylation is going on there. But to be more specific, that all cells that are metabolically active, they are constantly producing carbon dioxide. And carbon dioxide is an acid, right? When the carbon dioxide combines with water, water is present in all cells, it forms carbonic acid. And carbonic acid dissociates to hydrogen, that's proton, and that's the villain, like that's the evil, which is causing the fluctuations in the pH, and other is the bicarbonate. So this proton is threatening the pH to become lower, right? This proton is very dangerous to our body. We need to get rid of this. First we talk about that how our body is producing acid, and then we will talk about how our body maintains the pH within a narrow or normal range. Carbonic acid dissociates to proton. Every cell generates proton, right? In other words, we can say when every cell is producing carbon dioxide, so in other words we can say that every cell is producing proton. When sulfur containing amino acids like methionine, cysteine, and cysteine are broken down, the sulfuric acid is being produced. And when sulfuric acid undergoes reaction, it forms sulfates and protons. Again, protons are being formed, right? When phospholipids are broken down, phosphoric acid is produced. Again, this is also an acid, but on dissociation, it produces proton and phosphates, and again, proton are being produced, right? So you can see that in a lot of ways our body is constantly producing acids. But this bicarbonate, it's friendly because it's going to be acting as a buffer. We'll discuss it in a minute, okay? When we do strenuous exercise, our aerobic glycolysis shifts to anaerobic glycolysis. Okay, the pyruvate is converted to lactate by lactate dehydrogenase. And lactic acid is nothing but an acid, right? It's going to fluctuate our pH as well. When we use aspirin, there are salicylates, and aspirin in large doses can be converted to salicylic acid. It's again an acid. Methanol is converted to formic acid. Oxalic acid is formed by the ingestion of glycol, maybe ethylene glycol. Okay, ethylene glycol metabolism converts into oxalic acid, and again there is acid. When the patient is suffering from diabetic ketoacidosis, there is going to be production of acetoacetic acid. So you can see that a lot of acids are being produced. But what is the mechanism by which the body deals with such large production of acids? So let's discuss about that. First of all, what is the importance of normal pH? We know that enzymes that are proteins, and proteins are made up of peptide chains, and these chains are held together in specific configuration. Either they are primary configuration, okay, the secondary, tertiary, or quaternary. These configurations must be kept normal for the protein to be functioning, right? But these protons that are being produced constantly, they can change the configuration of the enzymes that are nothing but proteins. And you know that in our blood the pH is 7.4, and most of the enzymes, we are not discussing about the enzymes that work in the stomach. The stomach enzymes are working in acidic environment. They are used to it. Okay, they are not functioning in alkaline environment. Like pepsin, it's working, optimal pH is 2. It's very acidic. But most of the enzyme are functional pH of 7.4. It's normal. For example, this is the protein or this is the enzyme in orange. Okay, it's held together in specific configuration, and proton helps to keep that particular configuration. But if we add more protons, these additional protons can bind to that particular peptide or enzyme, and it can cause change in the configuration, and again the enzyme become dysfunctional. If we remove the protons from blood, if the blood become alkaline, these proteins that were containing protons originally, they will give they will donate their protons, okay, and again the configuration will gonna change. And in the patients with slight change in proton concentration, they are going to be dead because the enzymes are not functional, the metabolic system shuts down. And then comes the system of buffers that helps to maintains the particular pH in spite of changing the proton concentration or addition of bases or addition of acids. It doesn't matter, because buffer systems are there to protect the pH, to maintain the pH against the extracellular pH fluctuations. So these three are main mechanisms. Like buffer is the first line of defense. There are bicarbonate buffers, phosphate buffers, okay? Respiratory system is also gonna act as the buffer system. And third is the renal system. Kidney can secrete protons, can secrete bicarbonates, or it can reabsorb bicarbonate, it can reabsorb protons and potassium in order to maintain the alkalosis and acidosis, okay, in order to prevent that. So first of all, there is an experiment, it's very interesting. Action of buffers. The buffer main definition is that it resists the pH change. It does not allow the pH to change. First of all, the scientist was experimenting, right? First, we'll discuss about the experiment. There was a very unlucky dog. The dog extracellular fluid was 11.4 liters, okay? And the scientist took another container in which he took the same amount of distilled water solution. It means 11.4 liter distilled water solution is being taken in another container. So the unlucky dog was injected with 150 millimoles of HCl, and it's a huge quantity of HCl. When the dog was injected by the 150 millimoles of HCl, and he injected the same amount of HCl into the container. And as you know, the dog is a biological system and the container is non-biological. It's like a physical system made by man. Before injecting, both pH were kept 7.4. Dog, it's originally by God, it's 7.4, but the solution pH was also made 7.4 before injecting the HCl. But after, when injecting a large quantity of HCl, the distilled water solution dropped to 1.8. It's very logical because there are no buffer. But in dog, the pH only dropped to 7.14. It's a little change in the pH, although it's very lethal. This kind of change in pH is very lethal, but still there is enough like support or resistance to the pH that it does not drop to 1.8 as the distilled water. So from here, the concept of buffers arises that there are some molecules that are present in the biological system of dog that captured the hydrogen ions, that captured those extra hydrogen ions that were coming from the hydrochloric acid, and it resisted the pH change. These are the concept of buffers. Okay. I hope you understand the concept regarding the buffers. So let's talk about this example. It's a very interesting example as well. We are taking two containers, and in first container, which is A, there is no buffer system acting, right? And in the second container, which is B, there is buffer system. And buffer system is what, if we can draw the buffer system like this, okay? Buffer system has protons and it has like empty sites. When the pH is gonna increase, for example, if the pH of the blood is suspected to alkali, for example, NaOH, okay, so it's gonna donate these protons. The buffer is gonna donate these protons into the blood, and so the pH will not change as much, right? And if the pH is going to decrease, in which the protons are gonna increase in the blood, so this buffer system can take these extra protons, right? So you can see that this buffer has an unprotonated form, which is this form, and has a protonated form, which is this form. And a good buffer is that which has both the protonated form and the unprotonated form. So let's discuss about it. First of all, in container A, which where there was no buffer, when we add HCl, the HCl dissociates into protons and chloride, right? And this proton is going to change the pH of this solution because there is no buffer. But when we add the HCl into the same into another container with same amount of liquid, but this liquid or solution has a buffer system, okay? This HCl added, it also dissociates into protons and chloride, and these protons are then captured by the buffer system. These protons that were dissociated from HCl, these are captured by the buffer system. It means that the pH of the solution is not going to change. Okay, let's talk about alkali. When we add alkali to a non-buffering system, it will dissociate into sodium and hydroxyl ion, and this hydroxyl ion is going to act with the hydrogen already present in the solution, and it's going to make water. Okay, and again the pH of the solution is going to rise. There are already protons present inside the solution that were maintaining the pH, right? And when we add the alkali, this hydroxyl ion combines with the hydrogen ion and forms water. But in the container B, when NaOH is being added, it will dissociate to OH plus Na, and this OH will combine with the hydrogen ions present in the container to form water. But these protons, okay, these protons are helping the hydroxyl ion to form water, and this process is changing our pH. It's going to rise our pH, right? It's going to be alkalosis like thing, okay? And then the buffer system starts to donate its own protons in order to resist the pH fluctuation. It's gonna donate its own protons, right? And in this condition, it's gonna accept the extra protons. It's like donating something we have already and accepting that is extra. It's like a bank, when we have an extra money, we submit or we deposit the money in the bank, and when we are deprived of money, the bank, we can take when we can withdraw the money from the bank. Okay, it's like that. The buffer system has both hydrogen binding capacities and other like protonated form. So let's discuss about this. Buffer binds reversibly to protons. It means that they can add or remove the protons. They can add, it means that they have extra spaces to bind the protons, and they can donate the extra protons. So there are three types of buffers: extracellular buffers, intracellular, and urinary buffers. It's a very important equation in order to understand the concept regarding the buffer, that buffer can be in both forms. It can be in non-protonated form, in which this A minus form is non-protonated. Hydrogen is not being attached to A negative. It's non-protonated form of the buffer, or we can say it's the basic form of the buffer. And Ha is the protonated form of buffer, or we can say that it's acidic form of buffer. And those buffer solutions, or those systems in which the buffer has both the protonated form and non-protonated form, these are the best buffers. Okay, because when the hydrogens are more, when hydrogen amount is very very exceeding in the blood, so these buffer can take up the hydrogen, they can take the hydrogen, okay, and they can shift to Ha form, they can shift to protonated form. And when there is very low hydrogen present in the blood, or when there is alkali present in the blood, so this protonated form can donate its hydrogen and can be changed to non-protonated form. It's like that. Okay, and these are very important to maintain the physiological pH in response to acid base challenges. And in the next lecture we are going to discuss about in detail about Henderson-Hasselbalch equation. Okay, and we are going to derive it and some examples regarding it, some applications, right? So I will see you in the next video. Till then, goodbye.